are two professors. One is a mathematician and one is a physiologist. So they are both very interested into the idea of Braitenberg vehicle. And they have a lot of data in like the real recording of mouse, the chase of mouse, like seeking for audio sources. So they would like me to further develop my platform and to like encode some sort of ideal version of mouse traveling in space, like to find audio sources and to like compare that with their data.
Wow.
Mm-hmm.
It sounds pretty good. So does your school have a strong area? Is that a strong area for your school?
Sorry?
Is it like, do they have a lot of expertise in that area at your school?
Yes. So Pitt is like have a lot of faculties in neuroscience department. I think that Pitt's neuroscience department is the largest. I can't say it is the best, but it is the largest in the U.S.
Okay. Yeah. Definitely.
Yeah.
So I'm glad it went well. I don't know if Stefan is joining us today. I know he was at INCF. I don't know if he's still there or not. I haven't heard from him. I saw like some things about it, but I haven't heard anything from him.
So he like physically there, or
oh yeah, he gave like they had these demos where I guess it's like a poster session, but you have like your computer there and you can give a demo of the program, and you run through it for people. So we went over it. It was, he had like a 4-hour block where he had to people would come up to him, ask him about the demo. So he had a PowerPoint slideshow and he had his program open. So he just ran it for people. And so what is your vision for the program your project here that you're working on? You said that they were interested in it.
Yeah, they want me to firstly probably do something with the olfactory bulb. So they think that I can do more with like the afferent stuff, like what is after the olfactory bulb, and also they wanted me to think about using 2 sensors, because I was using just 1 sensor and 1 motor in the stuff I was doing during summer. So they wanted to see like what if there are 2 motors and 2 sensors, and probably I could. So there's a concept in animals called casting. So like they make very, very small movements in their in their nose. So like to seek the direction of the source, so that they can go toward that direction, rather than just like, because my implementation during the summer is like there's 1 sensor and to just like go forward if it's like, and go backward if it doesn't like. So if you're using casting, then you can see like some small like movement in dots, and then find ideal like source, ideal direction of the source, and then go toward it.
Good.
Yeah, so they want me to like firstly deal with this 3 stuff.
That sounds good. Yeah. So I don't know if Stefan is showing. Let me check and see if he's gonna show up. So we had also talked about use cases. We were talking about that earlier, a couple weeks ago, actually. And Stefan, I think in his slides, he proposed the use case for it's like a traveling salesman problem. So he was that's what he presented at INCF. So this idea that you have these agents and they can solve some problem like the traveling salesman problem but it would do it like how people do spatial integration. So it would be like where they'd use the cities as landmarks, and they'd, you know, sort of visit each city and come up with the shortest path.
I think it is a MP hard question, right?
Yeah, might be a little tough to do. Well, I mean, you know, in terms of setting up the environment, I don't know how long, you know, how hard it would be for the agents to do, but I mean, have you thought anything about like specific, well, other than the one you described to me? Oh, there's Jesse. Other than the one that you described to me, can you think of any other experiments that you'd like to do with your platform?
Experiments?
Well, I mean, like any experiments you think would be good to run in that platform?
I would say maybe, I don't know. So I was thinking about like the interaction between Braitenberg Vehicles because, yeah, but that's a kind of like a completely new area because I don't know.
Yeah.
Yeah, because I'm like still like thinking about making some progress for those, you know, just as those professors suggested. Yeah, do some improvements on those basic units.
Oh, okay. Yeah. So, hi Jesse, how are you?
Hi.
Oh yeah, I just yeah, I didn't really send anything out. Let's see. So you're we'll be able to talk much today. Oh, okay. So hi, we were just talking to Ziyi about his experiment or his talk at Pittsburgh, where he goes to school, and he said that it went pretty well, and they want him to develop the platform further, so that should work out pretty well. I mean, you know, I can't think of, I guess, you know, in the course of your specific field, there are probably questions that people are, you know, use animal models for, like certain experimental paradigms. I'm not quite sure what they would be, but I'm not that familiar with the field itself, but you know, there might be some, I guess what I mean by use case is like a very specific type of experiment that everyone does. So like in spatial cognition, they have the Morris Water maze, and it's a very specific setup. And then, you know, there might be other experiments where, like there are other experiments in learning and memory where they do the same type of thing, where they have like a radio maze, you know, where they train the animal, or, well, they usually do it in mice and rats, where they train them on a maze where they put food in one arm of the maze, and then, you know, train them on that and see if they go down that same arm every time, even if they take food out, or if the animal explores each arm and to see if, you know, where the food is. So I mean, things like that are you know, something that if people were going to use it, they'd say, oh, I can use it for this certain experiment. So I mean, that's just something that I don't know how useful it would, I don't know how many people would get a lot of use out of it. They could probably just set it up for their own purposes, but so yeah, I'll be happy to, you know, like help you with that if, you know, if you want to, if you're continuing on with the project. And yeah, I mean, Stefan, I think, is going to continue on. I don't know how much you know, time he's going to have for it, or how much time you're going to have for it, although it helps when you have some buy-in from your you know, university on developing it, or they're encouraging you. So so Jesse, yeah, I mean, I'll answer your Slack message. Everything seems fine with the message. I mean, you know, it's fine if you contribute to Twitter and all that. And, you know, maybe next week you can present a little bit about, you know, your progress or your ideas. I know we've been kind of talking about it. Like I said, you know, something like a PowerPoint presentation would be fine. Just something to lay out some ideas. And then, you know, we can talk about them a little bit more. So, I don't know if, I haven't gotten anything back from Stefan. Yeah, sounds good. I haven't gotten any, let me check again. Someone was added to BD Slack. Let's see. I don't know who that is. Let's check. I can't remember. I know someone might have joined. Oh, you know that was yeah, Aiden. So yeah, he's the other person I was telling you about with the machine learning group. So he's actually a mathematician. And he's interested in a different set of questions than what we're talking about here. So he actually wrote a blog post recently on, we were talking about, he's interested in neural organoids, which are these small, little populations of neurons that they can grow in culture. They end up being these little spheres that people are using to study like brain function maybe, or it's a pretty young field, so it's not like people have found a lot of applications for it, but they're able to grow, so they can grow neurons in culture, like a monolayer, which is like where the cells just grow on a single layer of medium. But then they also have these, you can grow these organoids or embryoids, so the neurons, they differentiate from stem cells and they form these clusters, and they're functionally kind of like a little brain. You know, they send action potentials between one another, and they're, you know, shaped sort of in a sphere, so they have some morphology. And his idea was to mathematically model some of this, you know, growth and how they might communicate. So we've been talking about that. It's pretty early. Right now he just proposed like a mathematical model for it.
For whole brain.
What was that?
For whole brain?
Well, it's just for these little organoids. So they're like really tiny balls of neurons. But like, you know, it's like how do you get from like a single stem cell, neural stem cell, and then the population expands, and then they form neurons, and then they connect. And so it's, you know, I mean, it's not really a brain, but it's like a little, well, they call it an organoid, which means like, you know, like a kind of a tiny organ that might be something that. So yeah, we've been talking about that. He put a blog post up. He was talking about collaborating with people, getting data for, you know, from different labs on like the growth conditions and the cells. I didn't really know what kind of data they have, but you know, it might be quite interesting. So that's pretty early on that.
I see.
Yeah. So, yeah, Ankit isn't able to make it either today. I don't know, he's got a test coming up, so. Let's see. Okay, yeah, just wanted to check the chat. I don't think Stefan is going to join us. Okay. Do you know of any modeling early neocortex development? Yeah, there's a lot of stuff on early development of the neocortex. That's kind of what the organoids are. Like a lot of the models that we were looking at for organoid development were kind of like, so in humans the neocortex is folded up, and that's kind of unique in mammals because in most, you know, it's sort of like this sheet that expands of this 6-layered cortex, and then, you know, in humans it gets so big that it folds up, and it folds up in other like primates, but not to the extent that it does in humans. And so, you know, a lot of the models of morphogenesis for like neurons is, you know, like how does it how do you get these, layers of neurons to form these shapes? And in humans, it's folded up so that you have you've seen the human brain, pictures of the human brain, and the outside looks like cauliflower or some sort of like ruffled surface, So that's the folding of that sheet of neurons. And, so the idea is, well, we can like look at models like that to see how they, fold. And they fold because they don't, they don't have, well, we think it's because they don't have enough space in the skull, but it's probably also because when it folds like that, it can perhaps, make longer distance connections between areas. If you had a sheet, you'd have like two neurons that were maybe on the edge of the sheet. If you fold it in different ways, even if you fold it into a tube, they have a shorter distance of connection. But also, if you fold it into folds, you get more opportunities for that. So, I mean, to answer your question though about papers, there are a lot of papers potentially on that. I'd have to go through and find some good ones for you though. Like usually I find reviews and things like that. So yeah, that's pretty good. You're looking at that as part of my interests. Okay. Yeah, I mean, so yeah, it's an interesting area, like early development. So like, in early development actually is, you're talking about like once the brain is formed, because there, you get like very early development, you get what they call the neural tube, which is where you get, sort of a flat sheet of neural cells, and then it folds into this tube, and then that tube becomes sort of the forebrain and the spinal cord and that, and then everything like kind of differentiates from there. So that's really early development, but I guess like early in terms of like cortical development would be what you're talking about more. Yeah. I mean, I think in terms of the BV stuff, it would be more functional than like morphological. So like, morphogenesis is, more about the cells making connections, although, well, I mean, I don't know how relevant it would be. I mean, it's really, I think the interesting thing here would be like just functional development. So like, how does connectivity happen? Although, there probably is some aspect of like, how the neurons are organized, sort of the shape that they're organized in, that makes a difference. So, I mean, that's something that we can talk about a little bit more later. I mean, if you were thinking of it in terms of like, say, if you had a bunch of like, all right, so say we had a vehicle that was square, and we wanted to say, well, we want to see if we can put neurons maybe in certain locations within the vehicle, in the brain, like inside the vehicle, would that make a difference in processing? So what I'm thinking of here is like, if you had some sensors and effectors, in the model, like you have in the book. So you have, effectors by the wheels and sensors up by the antenna, say. Then, you'd have like maybe a series of interneurons that were inside the vehicle. Okay, so if you say, okay, well, we'll put some interneurons in to see if it makes a difference in processing. But I don't want to put them just anywhere. I want to put them like maybe in a ring, or maybe I want to put them like, contralaterally. So, if you had like a antenna on the right side, you'd have an interneuron on the left side, and then that would map to the wheel on the right side, and vice versa. And then those two interneurons might be connected via a long-distance connection, like somehow. And so that might make a difference in terms of processing or not. There are a lot of ways that we can think about topology as making a difference in how information is processed. So, yeah, I mean, so the idea then would be like, it wouldn't be so much like, it wouldn't really be a direct analogy to human neural development, but it would be more like, say, like a small connectome in an insect or something, where you have, specific cells that do something, and, you have relays, say, like, between the forebrain and the limbs, and, the position of those interneurons maybe make a difference in terms of, subsequent connections and subsequent behaviors. So like in C. elegans, for example, which is, they have a very small connectome, relatively speaking. It's like 300 neurons or so. You have certain neurons that do things like locally and things that do things sort of globally in the network. You have like neurons in like they have a sensory area in the front. I guess it's chemoreceptive related, where they have like little neurons, up in the front of the body, that in the head, that process things locally, and then they also send information down to the body so that it can move. So they have like a number of different like localized populations of neurons, most of them being in the head of the worm, but also down in the other parts of the worm. They don't have a spinal cord, of course. They do have sort of this bilateral symmetry, so they have a they have a nervous system that goes up and down the body. But yeah, I mean, I think spatial relationships within the organism, that's actually pretty important in some ways, to see what what the process of neurons producing behaviors is. Yeah. So, any questions, Z, about that, or anything else you wanted to add?
Nope.
Okay.
Mm-hmm.
I don't think Stefan is going to join us today. So, so anything else you guys are interested in, want to talk about, or?
Yeah. So this semester I'm currently running 2 projects simultaneously. One is like the Braitenberg Vehicles stuff I talked about. Another is a completely new project, which is also do it for course credits for graduation. So that project is using genetic algorithm to simulate process of retina evolution. So in mammal, the retina develop a very interesting shape, which is like lateral inhibition.
Okay.
So we want to see like if that solution, if that structure of the retina is going to be the some sort of stable points or stuff that in the fitness or something, if there's some alternative solution that isn't exist and make sense.
So it's like using genetic algorithms to look at things like vision and assessing it's sort of the way it works.
Yeah.
Or, yeah. Well, that's interesting. Yeah. So is this like, are you using like off-the-shelf genetic algorithms? Hi, Stefan.
Hi, Stefan.
Are you using off-the-shelf algorithms for this, or are you, like, what is the thinking? Like, are they using something specialized for the task?
Sorry.
I mean, so people are wanting to do this. Are people using like some standard genetic algorithm, or are they trying to design an algorithm that's tailored to this problem?
I don't know, because that is like in very, very early stage.
Oh, okay. Yeah.
So the thing, I'm not only going to encode the network structure, I'm also going to like encode some sort of variables in like cell shape and the arrangement of cells. So there's a lot of things to Very.
Oh, wow, yeah. So that discussion we just had was kind of useful in that way. Yeah, it's interesting. We talked about, like, in one of my other groups, we talk about cell shape a lot. It's, you know, with neurons, of course, you have, like, processes that go out. And, you know, so you have that aspect of it. Also, you know, they have the gap junctions that they use for, you know, some connectivity as well. But yeah, like the cell shape is really fascinating, and it's like kind of it's both a very difficult area to kind of get your arms around, but also very like, you know, people have studied it. There are a lot of ways that people have studied it a lot, but still don't really understand why cells have certain shapes. But yeah, I mean, we can talk more about that too. So, Stefan, thanks for. How was? I guess the question is, how was INCF?
I wanted also to say sorry. It's just my laptop that I couldn't plug in for a pretty long time.
Oh.
Now I'm switched to handy, so it's fine. Should be okay. Can you hear me well? Is it fine, like the connectivity?
Can you turn it up a little bit, or?
You mean the microphone or what?
Yeah, microphone.
Like right like this. Is it better?
Yeah, it's better.
Okay, let's do it like this. So yeah, the NCF was pretty good. Like, I did hear a lot of useful stuff, and there were many interesting people from the field. Although I must say it was really little what was there to behavior, or maybe like, cognitive science as a whole. There were more like people analyzing data, so it was kind of like, very similar to bioinformatics, what it went about. So not modeling in the first place, but rather data analysis, some algorithms that are there for for neurobiological data. It was also little to psychology, or maybe, like stuff like that happens on the basis of what is in brain, but rather more about what is there in brain and what are the cells that are in brain. Maybe some models, but only for the connectivity and connectome and pyramidal cells, I remember. So yeah, it was just more on the side of like biological side of things, I would say. Therefore, I also didn't show my presentation to like a lot of people because there were not that many people who were interested in it.
Yeah. Well, that's
the point. Yeah.
Yeah, I didn't know what the audience would be like, but that's good to have that though, because you can use it again. People will want to see probably the technical details at some point, hopefully. There'll probably be other opportunities to present it, like, I don't know what, maybe locally at your university, like Ziyi was just telling me about, like, he gave a presentation in his department, and they were pretty interested in, what was going on there. But yeah, but I'm glad, you enjoyed it overall. I think it's, like, I think sometimes, if you just run the simulation for people, it's like, they get kind of interested in it. Where did you have people who were really, like, excited about it? Like, even if they didn't know?
Yeah, there was a woman who was coming from the, how's it called? This BCI field. So, brain-computer interface side of things. And she was really interested. Like, she said it's really cool stuff, although she didn't ask about technical details or where she can download it because it's not her field. Just as easy as that.
Yeah, well, you'll get some people who really like it and they don't really want, understand the technical details, and then, you get people. Oh yeah, okay. Jesse said someone did on Twitter ask about the thing. So I think there was someone who, like, on the INCF Twitter account, they asked about the project, and they said, where do we find more information? So, like, I don't know, like it was, I don't remember the name, I have it somewhere, but, so yeah, there are people who are probably, you know, a couple people were interested here and there. I mean, that's not unusual for conferences. Yeah, I mean, we can, yeah, we can maybe find out more about it. Like, I can maybe contact him if he's interested in learning more about it. But yeah, I think it's good to have that, like, presentation anyways, because you'll have maybe more opportunities to present on it.
Yeah, sure. Yeah. It's just about this one INCF meeting that it didn't bring us any substantial evidence or maybe audience in particular. You know.
Yeah.
So we have to look somewhere else. That's the point.
Well, yeah. So I mean, it's so yeah. I mean, a good practice to present a demo and to, you know. So, what the conference overall? Did you attend a lot of the sessions?
Yeah, I've attended all of them, and I really enjoyed it. So it was quite good.
Yeah, and yeah, I mean, you said it was kind of like oriented towards like informatics more, or behavior.
Yeah, but nevertheless, I liked it because, you know, you can get insights and more state-of-the-art insights from the field. And I enjoyed it nevertheless, although the presentation wasn't that successful. The talks that were given there, it just give you an idea on what's really happening there in neuroinformatics and what it's all about.
Yeah. So Jesse said there's an INC. The next INCF meeting is in Seattle next year. I might try to go to that one. Yeah, if it's convenient to go to, you know, that would be good. I don't remember. I think, was it last year they held it in Montreal or something? I don't know if they go between the U.S. and Europe, or if they have a European one and a U.S. one.
Yeah, so the experience of them is really to do this kind of things in America, as far as I've understood it. So yeah, this meeting in Europe was something exceptional. I think most of the years they do it in America.
Yeah, North America.
On the new land, yeah.
Yeah, because I think, Montreal is a big destination. Like, well, they have a lot of machine learning there, but they also have a number of people in INCF, and so Seattle, you know, they have a lot of people who do that sort of stuff. So that's good.
Yeah, probably.
It's good that they take it to Europe every once in a while, and that people there get a chance. So,
that was quite a nice opportunity.
Yeah.
To visit it in Europe, I mean.
So yeah, well, I'm glad everything went well. So what are your plans for the coming months on this project? Are you still interested in developing it, or what do you want to do with it?
Yeah, I would take a break a little bit, because, you know, right now I'm starting a new project in my lab, what's all about phages therapy and stuff. So, well, I haven't yet, we haven't yet decided the exact topic, but I think that's what's upcoming. So I will need this month to prepare for the project for the other one, if it's fine.
Yeah, it's fine.
Just because I wouldn't be able to, you know, host the both projects. And then maybe if this month is kind of fruitful and we've decided on what to do in the project with phages, I, of course, might be switching back more actively to the Braitenberg Vehicles project.
Yeah.
In the next upcoming months. Yeah. But, you know, I still nevertheless would like to attend meetings. Sorry once again that I was late today. And to just, you know, be in touch about what is happening in the community and stuff.
Yeah.
That would be awesome.
You're welcome. I mean, if you're not really doing anything like during the week or whatever, you're welcome to join the meetings. We always have a good talk about things. So yeah, I mean, you know, well, I mean, all I'm saying is I encourage you not to, you know, because what happens with GSoC projects sometimes is people put them down and then never pick them up again, and they just kind of let them sit. But, you know, I mean, and I think a lot of projects, sometimes it's not really something that you can continue on with. But this is like, I think there's a good base here for something, but I don't know, it's obviously you have to fit it into your schedule, so.
Yeah, we have to find this out, basically, so maybe, maybe just talking about it, and also more ideal work on what's could be done there. That's probably what the project needs now, as far as I can see.
Yeah. So actually, when you came in, Ziyi and I were talking about doing some project on with genetic algorithms. It's in his home department. So I don't know if you wanted to talk to Stefan about that, Ziyi.
Yeah, sure.
Yeah.
So this project is in a very, very new state. So I'm proposing that to using genetic algorithm to simulate the development of mammalian retina. So
sorry, of mammalian what? Once again?
Retina.
Just a retina. Oh, okay.
So in retina, there's like a very structural stuff called lateral inhibition. So it's pretty hard to explain it in some sentences, but you can imagine there's a center and surroundings, and surroundings has inhibitory connections to the center. So surrounding the center, so that increase the sharpness of the image. So we want to how mammals evolve those stuff. So that that is a very, very interesting structure that we evolve from the nature. And we want to use the genetic algorithm to simulate that process, and we want to see if there's any other alternative stable points during that process that is not nature. Which I think it is interesting, but I don't know, it's in very, very early stage. I'm supervised by a professor who is an expert in machine learning and neural networks. Yeah,
that basically that sounds really cool what you're doing out there. And do you simulate the structure of this retina in some sort of physical simulation? So does it have any environment and surroundings of it? Or is it?
No. Actually, I just get started. Actually, I'm gonna get to work next week. Start. So I just so this 2 weeks I'm currently actively talking with my professor about that. So probably first at very early first stage, I'm gonna practice genetic algorithm, and also to firstly make a very simple model of this center surrounding shape and lateral inhibition retina. Yeah.
Oh yeah, lateral inhibition. Now I see what you mean. Yeah, I've heard about that one as well. So basically, I think what I've understood from genetic algorithms, working with genetic algorithms, is that you can apply them in two types of projects or maybe approaches that you can use them in. So first one is, for instance, it's just random search equivalent, right? So you just overlook some idea of genes from the nature, and then you base an algorithm, a purely computational algorithm on it, right? So for instance, something like finding passwords, so where you have the example I've shown, or I don't know if I've shown this or not, but nevertheless, you have just some, say, 10 letter password, and you know which symbols have been used there, and you have kind of fitness, of various passwords has been told to you how close is your individual to the actual password, and you start kind of searching through all of this, various combinations of symbols to find the password. And so basically in this setting, there is nothing natural in it. It's just an algorithm. It's a way to perform a randomized search, right? So just genetic algorithm, it basically gives you the ability to divide your problem in blocks and then kind of maintain the blocks of the successful solution in this case, for instance, which is better than just random searching, because you don't have intermediate steps which you can be sure they are right. Yes, and the second setting is basically what we wanted to, or maybe still want to do in Braitenberg Vehicles projects, is where we kind of employ this idea about genetic processes in some kind of natural setting. And then we are talking about simulation more, we kind of try to simulate natural processes and the nature itself, or maybe just do a simple model, more so to say, of the nature, and then try to run this genetic algorithms as something natural, so to say. So you see, in the first, it's how I see it. So maybe, I don't know if it gives something or not, but the first paradigm is just purely computational one, I would say. And the second paradigm is using genetic algorithms as some natural process in some natural simulation. So that's why I asked whether your retina would have some physical space. So whether your retina would be something that would be put in context of the simulated nature, or it will be like in the first approach, just some placeholder for a good solution, and you will be trying genetic algorithms as purely computational paradigm, so purely as an algorithm.
So upon my conversation with the professor, I'm going to recover the lateral inhibition, especially the connection patterns in the horizontal cells. So horizontal cells is like interneurons that integrates the inhibitory signals from the surroundings and then project the center. So I want to use genetic algorithm to firstly just random connections of those interneurons and form that sort of standard surrounding shape. And also there are other parameters of horizontal cells, like the size of the horizontal cells, the shape, and the spatial arrangement of the horizontal cells and stuff like that. Yeah.
So you're still going to model kind of a system which involves this retina shapes.
Yeah.
Yeah. Okay, I see. So
I'm going to use Braitenberg vehicle because I feel like there's no need to use Braitenberg vehicle.
Pretty much. I agree with you.
Yeah. How can I develop sort of fitness function? Because because the retina, you cannot. I don't know. Because I'm still looking into the Braitenberg. Looking into the genetic algorithm. Function stuff.
Yeah. So basically, this question about fitness function is, I would say, the most important one, because all the other things, they're conserved across all of the genetic algorithms, more or less, right? So I have had troubles with representing what I want in the format where I can say, okay, there is my genes, and this is my chromosome, and now I kind of mutate my genes, So it was also pretty difficult to pick what you're mutating on, what your atom called gene, of which your solution is comprised, so to say. Because it's not straightforward. You have to divide. Ideally, you have to divide all of your options of, in your case, retina structure, in several similar length strings that can be mutated position-wise, right? So you have to have a text, so to say, a chromosome with genes as a representation of your retina, which is basically a structure in space, so to say. You have to be able to represent it as a number of set of genes, which you can then interchange. that's the first topic. It's pretty, like, it's not straightforward, but of course it's even more difficult. It's even more difficult to pick a fitness function. So the one where you say, okay, like, you know, this is closer to the solution or less close to the solution I want to have. And if you don't even know which solution you want to have, but you have to have, like, you have just some properties of the solution that you would like to have in your final solution, or maybe at least close to them, well, then you have to take these properties into account, like the properties that you know about final solutions. So for instance, I don't know, like I want my Braitenberg vehicle to have, let's say, 2 connections between neurons and all of the other neurons not connected, right? So if I would say, okay, that's something I would like to have in my solution, then I have to, it's not that I'm just saying it to my fitness function, okay, that's something you do to me, because fitness function doesn't have, like, it's not conscious. You have to think carefully about a mathematical term that would describe what you want to the fitness function. So for instance, in this case, I don't know, like, the first idea I have about, like, having just 2 edges, but in, you know, in some hard times I can allow for 3 edges if it's really that needed, because I maybe also have other constraints to my fitness function. And so I would kind of do, I would say, okay, like, all of the solutions that have, like, 1 plus minus 2 edges will receive, like, a 0.85 bonus to the fitness function or to the fitness value, and all the other ones will receive minus 0.15 bonus, or maybe, like, you know, price for that. Yeah, and so you see, it's not like that straightforward. So the fitness function, I would say, it's the thing you have to really design and ideally care about, and ideally do a really good model of what you want to become at the end in your solution, because only the fitness function defines what will your solution be.
Mm-hmm.
Yeah.
I would add, yeah, I mean, and you mentioned this, but the encodings are also, like, so you start with an encoding, which is, of course, how you take, like, the retina and you describe it as a series of genes, and, of course, binary values. I mean, we're talking about just a string, but also have that organized in a set of genes that you can then apply to the fitness function. So, like, Stefan and I have discussed this previously. You know, we've had some discussions about, like, the way that the strategies people use in GAs to organize these encodings. So sometimes people use something called modularity, or, like, these building blocks, where they take their system and they encode the features of it into some binary space. And so, you know, you might have, like, the retina cell, you know, one shape or another shape, different cell types on different genes, right? And then you might have, you know, functional differences on a different set of genes. So you'd have, like, a hierarchical set of genes where you had, like, you know, you have a gene that encodes, like, a cell type, and then under a single cell type, you might have a couple other genes that define, like, different functions or different shapes. And then, you know, you can then use that to arrive at a optimal solution by, you know, evolving specific parts of the system one at a time. And sometimes this is a way to do it, and sometimes it's not. It's just a strategy people use. But to just kind of, like, get a solution for one part, then get a solution for another part, and so on. And then, you know, you can find, like, these local optima, which are, like, in the parlance of GAs, is, like, you know, something that you want, but you also don't want to converge a global solution to a local optimum. You want a local optimum, you want a global optimum. It's kind of like what people worry about in, like, machine learning with solutions. What I can actually point you to, like, a primer on GAs too, if that's something you might want to have as a reference.
Sorry,
I have like a primer on genetic algorithms. So if you want me to send you that, I can do that. So you have it as a reference. Yeah, I'll send it to Jesse too. So,
cool.
And I mean, there are a lot of strategies. I think probably the fundamental thing is like the fitness function, because you're, again, like Stefan said, but there are also a lot of issues in encoding that are tricky, especially when you're dealing with like something like a retina where you have like a lot of different parts to it. It's not like a homogeneous system.
Yeah, so probably that was the suggestion from a professor that is using that is to code like a very fake static connections that to like process the output of the retina to a higher level and then to physics function.
Oh, okay, yeah.
Yeah, but the problem now becomes like how would that static connections be, and yeah.
Yeah. I mean, probably there will be a lot of. It won't be as straightforward as that. I mean, because then the thing of the fitness function is like, too, is you have like this, like you can always get like an optimum solution just by applying a fitness function, but is that like the global optimum, or is it like something that it's just like converging towards, and it's not necessarily a global optimum? It's pretty tricky. So it's, well, I wish you luck on it. I don't know. You have to give us, like, if there's some really interesting results, you'll have to give us an update. But yeah. So Jesse, you have anything to add on this? Okay. Yeah. Well, thanks for that discussion. That was pretty good. I wish you luck on that, Ziyi. And then, well, okay. So, if we don't, do we have anything else we want to discuss today?
Not for me.
Okay.
On my side.
Okay. All right. Well, if that's all we have for today, why don't we adjourn, and we'll talk next week. And if you have anything that you want to talk about in between now and then, we can talk on Slack. Okay.
All right.
Talk to you guys later.
Yeah.
Have a nice week.
Okay.
See ya.
Thanks, you too.
Thank you.